Hello and welcome back to the Biochemistry for Health Sciences channel. Today's video is going to be about cell division. Cell division is very active early on in life. For example, the one cell zygote, which is the egg fertilized by the sperm, keeps on dividing to form larger and larger structures and eventually forming the infant, and then the infant reaches puberty, and during all this time cell divisions, a lot of our cells in the body are dividing very actively. In an adult also we do find cells that are very actively dividing. As we mentioned in previous video, cells in an adult can be divided into non-dividing or permanent cells. These cells do not divide. Then we have stable or quiescent cells. These cells normally do not divide, but when stimulated, they can divide. And then we have these cells here called labile or continuously dividing cells. These are the cells that keep on dividing. They're very active in cell division. And examples of these labile or continuously dividing cells include the cells in the bone marrow that give rise to red blood cells as well as white blood cells. Then the cells in the cavity, such as the cells lining the GI cavity or the urinogenital cavity. And we also have continuously dividing cells in the skin. Now, understanding cell division and related topics such as nucleotide synthesis as well as DNA synthesis has several applications in the field of pathology and pharmacology. For example, cell division is a very important process to understand in order to understand the disease cancer, which is the number 2 killer after cardiovascular disease. Another pathology chapter that involves a lot of understanding of cell division is the process of wound healing, as well as the process of the immune system functioning, as well as infections. These are huge chapters that you will cover in pathology. And then in pharmacology, of course, understanding cell division is important to understand the drugs that are used for cancer as well as the side effects of these drugs. So besides the chemotherapy drugs, understanding cell division is also very important to understand the immunosuppressive drugs. These are drugs that prevent tissue rejection and also are used to treat autoimmune diseases. So understanding immunosuppressive drugs as well as their side effects requires a good understanding of cell division. The objective of cell division is a timely and accurate duplication of cellular contents and then its separation into 2 genetically identical daughter cells. So in this way, one cell can become 2 cells, and then this cell can divide to form another cell, and so on. Now, in order for a cell to divide to become 2 cells, it has to go through a cycle. This cycle is called the cell cycle. Now, the cell cycle consists of 2 parts. The first is called interphase, which is made up of the G1 phase. G stands for gap. And then we have the S phase, the synthesis phase. And then we have G2 phase. So G1, S, and G2 form what we call as the interphase. And during the interphase, the cellular content is duplicated, including the DNA of the cell. DNA duplication or DNA synthesis is called replication. The second part of the cell cycle is called the M phase. Now in the M phase we have mitosis. This is when the duplicated DNA is separated, and then we have cytokinesis where there is separation of cellular content into two daughter cells. So for a cell to form a new cell, it has to go through the G1 phase and then progress to the S phase and then progress to the G2 phase and eventually enter the M phase, and after completing the M phase, it will give rise to the new cell. Now most of our cells in the body are in a G0 state. This is called the quiescent state. This is basically a non-proliferative reversible state. So in order for this cell in a G0 state to divide, it has to first enter the cell cycle. Okay, it has to first enter the cell cycle, basically enter the G1 phase. Once it enters the G1 phase, then it can progress through the various phases of the cell cycle to produce a new cell. However, along the way to the cell cycle, there are several other things that can happen to this cell. For example, the cell can enter a state called senescence. This is basically an irreversible state. It cannot go back into the cell cycle. And in this state, is characterized by the presence of a lot of damaged macromolecules. And this is something what we see in AIDS-related pathology. The cell in the G1 state can also go through apoptosis. So this is programmed cell death. So in the G1 state, the cell can also die. And as we shall see in a few minutes, apoptosis is a very important protective mechanism against cancer. If the cell manages to go through G1 phase successfully and enter the S phase, once it's in the S phase, once again, it can also go through apoptosis or senescence. If it successfully goes through the S phase and enters the G2 phase, once again in the G2 phase, the cell could go through apoptosis or senescence. If the cell manages to go through all of these phases and enter the M phase, then in the M phase, the cell can still undergo different fates. For example, it can undergo apoptosis, or it can undergo what we call mitotic slippage. So in this case, what we see happening is the chromosomes that are duplicated, they are not separated. So the cell becomes, from a diploid cell, it becomes a tetraploid cell. This is not very common. It's a rare situation, but this can happen. And if the cell goes through all the phases successfully, giving rise to a new cell, a daughter cell, then this cell can continue dividing, go back to the G1 phase, or it can exit the cell cycle and get into the G0 phase. So let's look at the key steps that we see in cell division. We are going to use hepatectomy surgery as an example of cell division. In the clinic, hepatectomy or liver resection is sometimes done for removing liver tumors. So once a piece of the liver is removed, we'll see that the cells near the cut surface, these cells that are normally in their G0 phase, they will enter the cell cycle. Then these cells will go through the cell cycle, divide to form new cells, and these new cells will then replace the lost cells. And the liver is an organ that is very well known to regenerate itself when it loses its tissue, either through surgery or through disease. Now, the process of cell division is a very complex pathway that involves many proteins. It's a very highly regulated pathway. Inactive proteins will become active proteins, and after they carry out the function, will become inactivated again. And this is very important to prevent uncontrolled cell proliferation, which may lead to cancer. We shall only give a few examples of proteins, mainly to explain the key concepts of cell division. As mentioned earlier, cell division is closely associated with cancer, and recent research shows that cancer involves sustained proliferative signaling that causes excessive cell division, mainly due to the inability of these cells to exit the cell cycle as well as to undergo apoptosis. So we'll see these connections with cancer as we go through the steps of cell division, and these connections with cancer are shown in brown color. As mentioned earlier, several proteins are involved in cell division. Some of these proteins actually promote cell division. They are like the accelerator of an automobile. Other proteins inhibit cell division. So they are more like the brake of an automobile. So among all of these genes that make proteins that promote cell division, some of these genes, when they are mutated, we call this proto-oncogenes. So some of these genes called proto-oncogenes, when they're mutated, they give rise to what is called oncogenes. And these oncogenes will basically give rise to excessive amounts of proteins that promote cell division, or defective proteins, these proteins that cannot be regulated anymore. They keep on working all the time. So either case, either excess proteins or defective proteins is going to cause unregulated excessive cell division, and that's going to cause cancer. Similarly, among all the genes that make proteins which inhibit cell division, some of these genes, what we call tumor suppressor genes, if they are mutated, and that mutation has to happen in both copies of the gene, that will lead to the absence of these proteins that inhibit cell division, or defective protein. It cannot inhibit cell division anymore. It has lost its activity. So in either case, without the brake present, without the inhibition present, once again we get the same end result. Unregulated excess cell proliferation that will then can lead to cancer. So this is what we will see, mutations in the accelerator genes and or inhibitor genes, the brake genes, can both cause cancer. Okay, so let's go through the steps of cell division. So we are going to, as mentioned earlier, we're going to see how a cell in the liver, which is in a G0 state, will enter the cell cycle and divide. So in the first part, we'll talk about the secretion of signals called mitogens. When part of the liver is removed, the remaining hepatocytes, as well as other tissue cells, the macrophages, lymphocytes, and so on, which are facing the cut surface of the liver, they will secrete signals. Remember, we talked about signals and receptors under cell signaling. So these cells will secrete signals that are called mitogens. Now, examples of mitogens include transforming growth factor alpha, hepatocyte growth factor or HGF, and interleukin 6 or IL-6. So, for example, the macrophages, T lymphocytes, as well as hepatocytes, they will secrete TGF-alpha. Now, TGF-alpha shows autocrine activity in hepatocytes. That means it will bind to the same cell that secreted the signal. Other mitogens, such as HGF and IL-6, they have paracrine effects, which, if you remember, they bind to cells next to the cell that secreted the mitogen signal. So what are these mitogen signals going to do? They are basically going to bind to receptors on the remaining cells in the liver that faces the cut surface, and they're going to trigger various signal transduction pathways that are going to cause these cells to divide and replace the lost liver cells. So let's consider one example of how this mitogen works, and we'll look at this mitogen, TGF-alpha. So this signal binds a receptor called epidermal growth factor receptor, or EGFR, that is located on the plasma membrane of the hepatocytes. Now, EGFR, this receptor, EGFR, this is an enzyme-linked receptor. If you had forgotten what this is, we discussed it in a previous video under cell signaling. This is an enzyme-linked receptor that has intrinsic tyrosine kinase activity. So this is basically a receptor tyrosine kinase, or an RTK. And we talked about RTKs when we talked about the insulin receptor in video 7.3. So let's see how this signal triggers a cell signaling pathway. So in the first step, the TGF-alpha signal that is secreted by the cells facing the cut surface of the liver, these signals will start binding the receptors, EGFR. So, for example, this could be a hepatocyte. And these receptors, EGFR, will be bound by the signal. Now, when the signal binds the receptors, the two monomers of the receptor will dimerize, will come together in the presence of the signal. And when they come together and they're bound by the signal, they will change their shape. And when the shape of the receptors changes, that will expose the intrinsic tyrosine kinase activity of the receptor. So basically, the receptor now becomes an enzyme. Its enzymatic activity, its intrinsic tyrosine kinase activity is activated. The intrinsic tyrosine kinase activity of the receptor will phosphorylate itself, and the phosphorylated receptor will now trigger the signal transduction pathway called the MAPK pathway, which stands for mitogen activated protein kinase pathway. In this pathway, the phosphorylated receptor, the EGFR that is now rich in phosphate groups, will attract proteins such as GRB2, SOS, as well as RAS. So GRB2, SOS, and RAS will all be attracted to the phosphorylated receptor. Now when RAS is activated, it will activate other proteins such as RAF, MEK, as well as a mitogen activated protein kinase family protein called extracellular signal regulated kinase, or ERK. So now this signaling will lead to the activation of ERK. Now ERK will phosphorylate and activate several nuclear transcription factors such as Jun and FOS. So these activated transcription factors will now bind DNA, various genes in the DNA, and they will stimulate the transcription of these genes, and increased transcription of these genes will lead to the increased synthesis of a variety of proteins that are required for cell division. And examples of these proteins we'll see in a few seconds. One of the most important proteins that are stimulated, that are increased, is these proteins called cyclins. These are proteins that activate enzymes called cyclin-dependent kinases, or CDKs. And then there are many other proteins such as the E2F family of proteins. So all of these proteins now will start increasing in the hepatocyte. Now mutations involving the mitogen signaling genes are common in cancer. So for example, the TGF-alpha gene could become an oncogene, and that could lead to excess mitogen signals. So you have a large amount of these signals that can lead to cancer. We could also have the EGFR gene, which could become an oncogene. So this receptor EGFR could also become an oncogene. In that case, you can get excess mitogen receptors. So you can have too much of these mitogen receptors, and that can lead to cancer. Or you could have defective mitogen receptors. Okay, so in these defective mitogen receptors, they are basically a dimer, and they're activated even when the signal is not bound. So if they're activated all the time, even without the signal, that is going to cause the cell to synthesize all of these proteins necessary for cell division, and the cell is going to keep on dividing, and this will increase the possibility of cancer. So besides problems with the signal or the receptor, one could have problems with the other proteins that are involved in cell signaling. For example, it is known that the overexpressed GRB2 is seen in several cancers. SOS gene could become an oncogene. The RAS, the RAF genes could all become oncogenes, and including the downstream proteins. Several of these proteins involved downstream. They can all also lead to cancer if they are found in excess amounts, or they cannot be regulated. They are defective. So now this cell has several of these proteins that are necessary for cell division, and these proteins were made in response to mitogen signals. In the second part, now we'll see that the cell will now enter the cell cycle, and there is a protein called cyclin D that starts accumulating, and this protein will help the cell enter the cell cycle and eventually progress to the G1 phase. So let's look at it. Now this protein cyclin D starts accumulating in the cell because of the response to the mitogens. Cyclin D binds CDK4 as well as CDK6. And this binding of cyclin D to the CDKs will activate the CDKs, and this will set the stage to prime the cell to enter the G1 phase. So now the G0 cell will enter the G1 phase, and as long as these protein cycling D is found in high amounts, this cell cannot go back into the G0 phase. It simply enters and then it progresses through the G1 phase. So it's not surprising that genes for cell entry, when they are mutated, are seen in several cancers. So for example, cyclin D1 is overexpressed in many cancers. The CDKs, such as the CDK4, CDK5, CDK6, these genes can also become oncogenes and become overexpressed, and we see that in several cancers. So once the cell is in G1 phase, it grows in size as it synthesizes more and more proteins as well as several organelles, because remember, this cell is going to form a new cell, so it has to get bigger and bigger. Now in part 3, we are going to look at this very critical phase of the cell cycle called the G1S checkpoint. So basically, in summary, what this checkpoint does is, before the chromosomes are copied in the S phase, we will see sensor proteins going around and checking for any damaged DNA. And if there is damaged DNA that is detected, we will see that the cell cycle will be arrested. So the cell cannot progress further. The cell cycle will be arrested by what we call CDK inhibitors. And then we'll also see that the DNA that is damaged will be repaired by DNA repair proteins. So let's look at this part 3, the G1S checkpoint. So as mentioned, the G1S checkpoint checks for DNA integrity, making sure there is no DNA damage before the cell enters the S phase, because in the S phase, the DNA will be copied. So we don't want to copy damaged DNA. So what we have is, we have sensor protein complex. This protein complex will sense or will detect DNA damage. And one example is this complex called mRN, which consists of the protein MRE11, RAD50, and NBS1. So this protein will go around the DNA and check for any DNA damage, such as a double-stranded break and so on. If it does detect a damaged DNA, then the mRN protein will then bind and activate another protein called ATM. ATM stands for ataxia telangiectasia mutated, ATM protein. So now the mRN has detected DNA damage. It will now bind and activate this protein called ATM. Now ATM will now phosphorylate and activate a serine threonine kinase protein called checkpoint kinase 2 or CHK2. So ATM will now activate CHK2. Now CHK2 will then phosphorylate and activate a transcription factor called P53. And the activated P53 will now increase the transcription of the P21 gene. So as the transcription of the P21 gene increases, we get more and more and more of this protein called P21. Protein P21 is a CDK inhibitor, also called a CDKI. It should be noted that there are several CDKIs. Other CDKIs examples include P16, P15, P18, 19, 27, 57, and so on. So we have a lot of P21 because the gene for P21 is being actively transcribed. So we're going to have a lot of P21. What does P21 do? This protein P21 will bind and inhibit cyclin-dependent kinase 2, CDK2. So the P21 will bind CDK2, and CDK2 is necessary to progress to the G1 phase. So when CDK2 is bound and inhibited, the cycle will stop. The cell cannot proceed any further. The cell is arrested in the G1 phase. So the cell is prevented from entering the S phase. Now, P53 not only increases the transcription of P21, it also increases the transcription of several DNA repair protein genes. And that leads to the production of all of these DNA repair proteins. And now the damaged DNA will be repaired by the repair protein. So while the cell is arrested in G1 stage, it's not proceeding any further, repair happens. The damaged DNA is being repaired. Now let's look at part 4. What if the DNA damage is too severe? It cannot be repaired. Now, if it cannot be repaired, we will see what does P53 do. P53 will now stimulate the pro-apoptotic genes. Okay, this will lead to the production of proteins that will make the cell undergo programmed cell death or apoptosis. So if the DNA damage is too severe, it cannot be repaired. This protein P53 not only directly stimulate apoptosis under the right conditions, it also increases the transcription of these pro-apoptosis genes. When we'll talk about apoptosis in a future video. So examples of these proteins that are increased include the Bex proteins, the Puma proteins, and the Fas receptor proteins. And all of these pro-apoptotic proteins are going to cause the cell to undergo cell death by apoptosis. Okay, and this is very important because the DNA cannot be damaged, so the cell basically is killed off so that we don't make a copy of a cell that has damaged DNA. Otherwise, there's an increased chance of getting cancer. So you can see the importance of this P53 protein. Not only does it arrest the cell division in the G1 phase, it also helps in DNA repair. And if DNA repair is not possible, then it kills the cell by apoptosis. It's not surprising that the P53 gene is sometimes called the guardian of the genome. It prevents division of cells with damaged DNA during the G1 phase. It helps the cell to decide whether to enter the S phase, exit the cell cycle, or die by apoptosis. Now, although P53 is considered a tumor suppressor gene, there have been cases mutated forms of the gene can actually gain new pro-oncogenic activities. As the guardian of the genome, it's not surprising that mutations in the P53 gene are among the most common that are found in various cancers. And we see that in almost 50% of human cancers, including breast, lung, as well as colon cancer. Okay, part 5. What if there is no DNA damage or the DNA damage is successfully repaired by the repair proteins? Okay, so if all this is possible, then now the cell will enter the S phase. Okay, so if everything is fine in the G1, cell enters the S phase. However, to enter the S phase, it requires this E2F dependent transcription, which is necessary to make cyclins E and A. Cyclin E and cyclin A are very important to progress to the S phase. So let's look at this. So what is this E2F dependent transcription that is so important for the cell to enter the S phase? So this E2F dependent transcriptional network is a network that is activated in the G1 phase. It drives the cell to enter the S phase. This network becomes inactivated during the S phase. The E2F network includes many genes that encode proteins in cell cycle, in replication control, in growth, as well as in DNA protection. These E2F transcriptional factors are several. There is E2F1, E2F2, E2F3, 4, 5, 6, 7, 8. So besides the transcriptional factors, E2F1 to 8, they also have very important co-regulators. So these co-regulators regulate these transcription factors. These are sometimes called pocket proteins. And one very important example of a pocket protein that regulates E2F, we'll talk about this, is retinoblastoma protein PRB. Other pocket proteins include P107, P130, and so on. Now, these E2F transcriptional factors will only become active when they are released from their co-regulators. Okay, so co-regulators basically block the E2F activity, but when the co-regulators are released, then these E2F proteins become active. Some of these E2F proteins are activator transcription factors, while other E2F proteins are repressor transcription factors. So some of them actually increase the transcription of certain genes, others decrease the transcription of certain genes. Due to the importance of E2F in the cell cycle, it's not surprising that several of these E2F genes can be oncogenes and associated with cancer. So, for example, the E2F1, the 3A, 4, 8, which is seen in liver cancer, all of these are associated in various cancers. The E2F1 can also act as a tumor suppressor gene besides an oncogene. So as mentioned earlier, E2F proteins will only become active when they are released from their co-regulators. So let's look at an example of a co-regulator, PRB, and how this co-regulator is released so that the E2F protein can become active. Now PRB, the protein PRB is encoded by the gene called RB1 gene, which stands for retinoblastoma 1 gene. And this gene is located in chromosome 13 on the Q arm. Now, in the absence of any mitogenic signal, this PRB, this protein exists in an active hypophosphorylated form. So this protein has very few phosphate groups. So in the hypophosphorylated form, this protein is active, and being active, it will bind and inhibit E2F transcription. So by inhibiting E2F transcription, there's not enough cyclin E and cyclin A, and the cell cannot progress any further. The cell is arrested. Therefore, we can see PRB is an inhibitor of cell division. And this is shown in this diagram. PRB bound to the E2F transcription factors, and the transcription factors cannot work as transcription factors because they are inhibited. And without this E2F transcription, there is no cyclin E present, no cyclin A present, or their concentrations is very low. So these enzymes are not activated, and that will lead to inability to enter the S phase. So how does the cell stop this PRB from working so that the cell can start entering the S phase? So let's look at this. So what happens is the cyclin D that is bound to CDK4/6, this enzyme will phosphorylate the PRB. Okay, and when this PRB becomes phosphorated, it becomes inactive. And when it's inactive, it doesn't bind E2F anymore. And when it doesn't bind E2F anymore, the E2F will transcribe the genes, and we have cyclin A and cyclin E, and this will allow the cell to enter the S phase. Recent research also shows that cyclin E CDK2 may also be responsible in phosphorylating and inactivating PRB so that the cell can enter the S phase. Cyclin E has been seen to work as an oncogene in certain cancers. As you can see, cyclin E plays an important role in cell cycle. There is a positive feedback loop between cyclin E CDK2 as well as the E2F dependent transcription. So the cyclin E CDK2 activates E2F dependent transcription. As this becomes more active, it will produce more cyclin E, which will activate CDK2 that goes back and activates the E2F dependent transcription. Okay, so now the cell has overcome the action of PRB, the pocket protein PRB, and now the cell is able to enter the S phase. So let's look at part 6, the S or the synthesis phase. So cyclin E as well as cyclin A, these two cyclins that activate CDK2 play a very important role for the cell to progress through the synthesis or the S phase. Now there is a protein called APCC, which stands for anaphase promoting complex cyclosome, APCC. And when this protein is complex with CDH1, so APC complex with CDH1, this protein will stimulate the destruction of cyclin A by the proteasome. So, and you notice that cyclin A is required for S phase progression. So to stop this cyclin A destruction, there are two mechanisms that we know of. One is cyclin E CDK2. This complex, this enzyme will phosphorylate and inhibit this APCC CDH1. And then there's also an inhibitor called early mitotic inhibitor or EM11. And this inhibitor also inhibits this protein. So with the help of these two mechanisms, this protein is suppressed during the S phase, and that will lead to increased amounts of cyclin A, and that will help the cell progress through the S phase. These CDKs, they make sure that the genome is replicated at the right time during the S phase, and only once in the cell cycle, and that is only in the S phase. So due to their roles in the cell cycle, it's not surprising that APCC as well as EMI1 can act as oncogenes. We see that in certain cancers. APCC, when it's bound to CDCH1, to the CDH1, this acts as a tumor suppressor gene. So once again, in some cancers, we can see problems with these proteins. So now the cell goes to the S phase. During this time, active nucleotide synthesis occurs because to make DNA, you need nucleotides. The DNA is copied, the chromosomes are duplicated. This copying of the DNA starts from replication origins. So throughout the DNA, the actual synthesis or the replication starts from certain points in the DNA that we call replication origins. And we'll talk about DNA replication in a future video. DNA replication happens only during the S phase, once in a cell cycle. To prevent reinitiation of DNA replication, the origin licensing occurs in G1 phase, while the origin firing occurs in the S phase. So what is this licensing and firing? Licensing is basically loading of the inactive helicase complex called the MCM helicase complex onto the origins of replication. Okay, this helicase we'll see later is required to unwind the two strands of DNA in order to copy the DNA. In firing, the DNA replication is initiated by formation of a transient pre-initiation complex and then subsequent activation of the helicase. So this loading of the MCM helicase, onto the origins of replication, this happens in the G1 phase, while the actual DNA replication initiation, that happens in the S phase. So the firing of some of these licensed origins happen in the S phase. So in the S phase, all the 46 chromosomes will be replicated. Each chromosome will end up with two identical sister chromatids. Besides chromosome replication, the centriole duplication will start at the G1S transition phase, forming procentrioles, which are daughter centrioles, from each mother centriole. These procentrioles will continue to elongate during the S phase as well as during the G2 phase. Okay, as we mentioned earlier, we will discuss more about nucleotide synthesis as well as DNA synthesis, both of which have to happen in the S phase so that the DNA is copied. We'll talk about this in future videos. A few words about replication stress. Okay, so this is not a DNA damage, this is replication stress. So replication stress is checked. There's a replication stress checkpoint. So what does this replication stress checkpoint do? It monitors the progression of DNA replication forks. So when DNA is synthesized, we have replication forks formed from the origin of replication. So we have many replication forks. And we'll talk about this during DNA synthesis. So these replication forks will travel as DNA is copied. Now, any condition that slows down the progress of this replication fork or any condition that stops the progression of this replication fork, this is called replication stress. So as the DNA is copied, these replication forks may encounter obstacles that can slow down the progression of these forks, or they can actually stop DNA replication. And examples of these obstacles include exposed single-stranded DNA, unusual DNA structures such as G quadruplexes, as well as imbalances in nucleotides. For example, remember that for every A there has to be T nucleotides, for every G there has to be C nucleotides, and so on. And if there is imbalance of these nucleotides, that can also lead to replication stress. So what happens if there is replication stress? So what does this replication checkpoint do in case there is replication stress? Any replication stress at the stalled replication fork is detected by a protein called replication protein A, or RPA. So RPA will detect any replication stress, and then RPA will recruit and activate another protein called ATR, which is ATM and RAD3 related protein. Now ATR will then go and phosphorylate and activate the protein CHK1. CHK1 then will phosphorylate the phosphatase enzyme called cell division cycle 25, or CDC25. Now CDC25 will then be degraded by the proteasome. Now CDC25 is a strong activator of CDK2 as well as CDK1. So without CDC25, this CDK2 activity slows down and the cycle will be arrested in the S phase. So the replication stress checkpoint plays a very important has several important functions. It blocks late origin firings, it stalls replication forks, it stabilizes the stalled replication forks, and this checkpoint response makes sure that all DNA is replicated by preventing replication at new sites, slowing down replication, allowing the replication forks to only continue once the obstacle is corrected. Now, if there is prolonged stalled replication fork, the fork doesn't move for a long time, that can lead to the destruction of the fork. The fork collapse and its dissociation of its components, and can even lead to double-stranded DNA breaks, and this will trigger the DNA damage repair system, which is different from the replication stress system. And now if there is no replication stress, or the replication stress has been corrected, all the DNA will be copied, and now the cell is ready to enter the G2 phase. So let's look at part 7, which is the G2 phase. In the G2 phase, each procentriole elongates, matures to form centrioles, resulting in 2 centrosomes. Remember, each centrosome has 2 centrioles. The cell will now double its size as it makes new proteins and other biomolecules which are required for the newly formed cell. And once again, once it reaches a certain point called the G2M checkpoint, the cell will now check for DNA integrity, make sure that all the DNA, including the newly made DNA, is free from any DNA damage. If there is no DNA damage, then the cell will progress to the G2 phase and enter the M phase. However, if there is damage, DNA damage, then the cell will be arrested in the G2 phase, and the DNA damage will have to be repaired. And once again, if the DNA damage doesn't get repaired, the cell will undergo apoptosis. So let's look at this part here in case there is DNA damage. If there is DNA damage in the G2 phase, both of these proteins, ATM as well as ATR, will become active and they will phosphorylate CHK1 as well as CHK2. The CHK1 and 2 then will phosphorylate the CDC25. CDC25 will be phosphorylated, and this CDC25 will be degraded by the proteasome. And without CDC25, remember CDC25 is an activator of these enzymes, CDK1. So CDC25 is an activator of CDK1. Without CDC25, CDK1 activity goes down, and when CDK activity goes down, the G2 phase is arrested. The cell basically is arrested in the G2 phase. So besides arrest at G2 phase, there is also stimulation of the p53 pathway. And with the p53 pathway, remember what happens? There is DNA repair. So if the DNA is repaired, then the cell will continue into the M phase. But if there is no DNA repair, or DNA repair cannot be done, is too severe, then the p53 will also stimulate apoptosis. In that case, the cell will die in the G2 phase. So if the DNA damage was repaired or there's no DNA damage, now the cell will enter mitosis. So let's look at part 8, mitosis. Now to enter mitosis, CDK1 plays a very important role. CDK1 is only active when it's bound by cyclin B or cyclin A. Now there are kinases such as WEE1 as well as MYT1, which will phosphorylate and inhibit CDK1, preventing it from entering mitosis. The protein phosphatase CDC25 removes these phosphates from CDK1 and activates CDK1. So CDC25 actually helps the cell enter mitosis. So a balance between V1 MYT1 that inhibits CDK1 and CDC25 that activates CDK1 is a very important balance that determines when the cell enters mitosis. And this balance is regulated by multiple pathways that makes it a very complex system. So due to their importance, it's not surprising that MYT1, CDC25, and V1 can all act as oncogenes, and we see that in certain cancers. So why is CDK1 very important? CDK1, this enzyme, cyclin-dependent kinase 1, will phosphorylate many substrates during mitosis. And it also phosphorylates and activates mitotic kinases enzymes such as PLK1, Aurora A, Aurora B. So and when these enzymes are phosphorylated, they become activated, and they in turn will go and phosphorylate many substrates. So CDK1 as well as these enzymes end up phosphorylating many proteins and many substrates during mitosis. Aurora A and C have been seen working as oncogenes in some cancers. So let's see some of the effects of this cyclin B that is activating the enzyme CDK1 in mitosis. The cyclin B CDK1 is important in the cytoplasm for cell rounding as well as centrosome separation in the nucleus. the BCDK1 activity triggers chromosome compaction into discrete structures, what we call condensation. It triggers the activation of APCC, and this is important later for targeted cyclin degradation in order to exit mitosis. It also triggers the disintegration of the nucleolus, and it also makes the nuclear envelope permeable. During the prometaphase part of the mitosis, the nuclear membrane is now all gone. Without the nuclear membrane, the nuclear and the cytoplasmic contents are mixed. The microtubules form and attach to the kinetochores of the condensed chromosomes to assemble the mitotic spindle, and there is cyclin A degradation. So during this time, this cyclin A here becomes ubiquitinated by the APC complex to CDC20. So this protein will cause cyclin A destruction by the 26S proteasome. So without cyclin A, some of the CDK would not be activated. However, cyclin B is still in high concentration, high activity, and that will keep on stimulating CDK1 activity. Once again, due to the important role of APCC, we do see APCC acting as an oncogene when it's bound to CDC20 in this case, or as a tumor suppressor gene if it's bound to CDCH1 as we talked about earlier. During metaphase, there is chromosome alignment along the center of the cell, and then there's a very important checkpoint called the spindle assembly checkpoint. The spindle assembly checkpoint ensures that the chromosomes are divided equally between the two cells. Errors may include uncaptured chromosome by the spindle, two sister chromatids attached to the same spindle pole. This spindle assembly checkpoint is made up of a multi-protein complex. It binds any kinetochores that are not attached to microtubules after phosphorylation by Aurora B as well as CDK1. This spindle assembly checkpoint, or SAC, will catalyze the formation of a diffusible mitotic complex made up of various proteins like MAD2, BUBR1, CDC20, and this complex will inhibit ABC-CDC20 and prevent the initiation of anaphase. After all the kinetochores are attached and oriented correctly, SAC activity will decrease and the mitotic checkpoint complex will disassemble. Even one unattached kinetochore will maintain the APCC inhibition and will cause mitotic phase arrest. Cells cannot exist mitosis until the SAC is satisfied. If chromosome orientation is not resolved, the cell will undergo apoptosis or exit the M phase without chromosome segregation. In that case, we will get a tetraploid cell. During anaphase, the anaphase promoting complex cyclosome, APCC, which is now complex with CDC20, will trigger the breaking down of the protein cohesin that holds the sister chromatids together, thereby allowing the separation. The APCC will also trigger the mitotic spindles to shorten and pull the chromatids towards opposite ends of the pole, and it will also help in the formation of the ectomyosin contractile ring to help cytokinesis that happens later on. During telophase, the chromosomes arrive at opposite ends of the cell and decondense. The nuclear envelope begins to form around each set of chromosomes, and the mitotic spindles break down. And in cytokinesis, the actomyosin, made up of actin-myosin contractile ring, pinches the cell membrane inwards until two daughter cells form. For mitotic exit and completion of the cell cycle, the cyclin B is ubiquitinated by ABC CDC20, and the cyclin B is then degraded by the 26S proteasome. This destruction of cyclin B abolishes the CDK1 activity, and that will lead to mitotic exit and completion of the cell cycle. The protein phosphatases will dephosphorate various substrates, reversing CDK1 activity. If there is enough cyclin D, it will stimulate CDK4/6, and that will allow the cell to once again enter the cell cycle. Otherwise, the cell will exit the cell cycle and enter into the G0 phase. So this is basically the cell cycle and how a cell divides into two. We made this into one long video rather than several short videos. That's because we want you to make connections, and for healthcare professionals, in order to apply biochemistry and especially cell cycle to cancer, it's very important to make connections, and connections are best made in one long video. To summarize the connections with cancer, we see that the most common gene mutations that we see in cancer are associated with the mitogen genes, those responsible for the signals, receptors, and downstream proteins, as well as the genes that control cell cycle exit and the DNA damage checkpoints, for example, the p53 tumor gene that's found in many cancers, and the cell cycle entry genes, which depends on E2F-dependent transcription, and this is also commonly seen in cancers. Mutations in genes involved in replication stress as well as spindle assembly checkpoints are rarely seen in cancers. Hopefully this video gives you an idea of cell division and its association with cancer. And due to the complexity of cell division, we can see why treating cancer is a very challenging thing to do. Until the next video, thank you for supporting this channel. Have a wonderful day, and hopefully we'll see you soon.